The DNA acts as a genetic material and encodes all the information required to encode a protein. The specific region of DNA that encodes the genetic information is called a gene, and there can be several genes in the genome. For example, E. coli has roughly 4,500 genes, while humans have 30,000 genes. It is usually by the process of transcription, the genetic information in the DNA flows in RNA, also known as the messenger RNA or mRNA. The information contained in mRNA is further used by the translational machinery to make proteins. This flow of information from DNA to RNA and protein is also known as central dogma of molecular biology. This was given by Francis Crick in the year 1958. The genetic code. Genetic code is a particular sequence of nucleotide that tells which amino acids are to be linked together to form a protein. The genetic code consists of 64 triplets of nucleotides that codes for 20 amino acids. Each of these triplets are known as codons. Now, as there are 64 codons and only 20 amino acids, a single amino acid can be coded by more than one codon. This property of genetic code is known as degeneracy. For example, amino acid proline can be coded by CCU, CCC, CCA, and CCG. However, a single codon will always code for a single amino acid. For example, CCU always codes for proline. AUG always codes for methionine. This property of genetic code is called non-ambiguity. The genetic code is non-overlapping, which means after reading one triplet, the reading frame shifts over the other three nucleotides. The genetic code is commaless, which means there is no extra nucleotide between the codons. The genetic code has start and stop codons. AUG is usually a start codon that codes for methionine, and stop codons are UAA, UAG, and UGAA. The stop codons are also known as nonsense codons as they do not code for any amino acid. The genetic code is nearly universal, which means it is same in humans, bacteria, plants, and amphibians. However, there are some exceptions in mitochondrial genome, single-cell eukaryotes, and some bacteria. The first two bases of the genetic code are same, while at the third position, the base may wobble.